Okay, so we've talked a ridiculous amount about DNA, this double helix shaped molecule that's present in every nucleus in your cells. I think I've mentioned that if we could take all of the DNA out of one of your cells and untwist it and untangle it and lay it end to end, you'd have about 6 feet in every cell. And we know that it's important for heredity. We know that it's the transforming principle. We know it's one of two things that your chromosomes are made of, the other one of which is proteins. But to understand how it does what it does, we have to bring in a second type of nucleic acid called ribonucleic acid, or RNA. These first few slides are a repeat of what we looked at before break, but it's been a while. I figured it wouldn't be a bad idea to review. So RNA is a polymer of nucleotides like DNA. The big differences are that RNA is typically single-stranded instead of double-stranded. It's not a double helix. Sometimes, in fact, pretty often, a single strand might end up bonding with itself, and you'll end up with a loop, or you might end up with a little part of it that's kind of twisted. But by and large, it doesn't form long-term stable double helixes. The nucleotides contain a sugar that is called ribose, not deoxyribose, and one slightly different base called uracil instead of the base thymine. So again, on the left, that's a nucleotide that you would find in DNA. It's got the sugar deoxyribose. Remember, if you forgot, that every nucleotide consists of 3 parts. The phosphate group is an atom of phosphorus with some oxygen stuck to it. There's a simple sugar, which is shaded here in green, and then one of 4 possible ring-shaped molecules with nitrogen in them called bases. So in DNA, the sugar is missing an oxygen that would go right here. So that sugar is deoxyribose, and the nucleic acid made with it is deoxyribonucleic acid. And then on the right, this RNA nucleotide does have that little missing oxygen right there. So we don't call it deoxyribose, we call it ribose. And the base is a little bit different, and it's not called thymine, it's called uracil, which until I was a biology major, I always thought uracil was a brand of battery that had like copper tops, but I guess not. So what happens? I compared this to going to the library and finding a recipe in a book that's in the reference section. These are the books that you cannot take out of the library. You can browse them, but they don't leave the library. What you could do would be to make a photocopy of the information and take it out of the library and use it to cook a dish in your kitchen. That's the best metaphor I can think of for what happens here. The first step is called transcription. And in transcription, a length of DNA unzips, and RNA nucleotides come in, and they're assembled to make a strand that's complementary to one of the DNA strands. And it follows the familiar pairing rules. If you have an A on the DNA strand, it will bond with a U on the RNA strand. And a T on DNA will bond with an A on the RNA, and G and C will bond with C and G. So it's just like we've covered, except that if it's RNA, it has uracil everywhere you'd expect thymine. When that RNA strand is completed, it peels off and leaves the nucleus and goes out to where it's needed. And that RNA strand is specifically called messenger RNA. So if you found a recipe in a library book and you made a photocopy of it, the copy that you carried out in your pocket would be messenger RNA. So there's a diagram of what it looks like. You can see the DNA double helix in the center, and then in the middle, there's a giant purple blob. That's actually an enzyme called RNA polymerase that does the actual work of assembling RNA nucleotides into a strand of RNA. And so if the DNA strand says, I'm just going to make something up, TAA, TACCGAT, if one of the DNA strands, which by the way is called the coding strand, the template strand, if the template strand of DNA has that sequence of nucleotides, then as the RNA molecule nucleotides come in, they're going to form what? Well, there's a T right here. So the first RNA that comes in is going to be an A. There's an A right here. So the RNA nucleotide that can bond here is not a T, it's a U. And then this will be another U. And this will be another A. And this will be another U. And then this will be a G and another G and a C and a U and an A, and so on. So again, it's the same base pair rules that you've learned, with the wrinkle that RNA doesn't use thymine. It uses uracil in the place of thymine. By the way, little momentary ecology thing. My son and I went for a walk in the nature reserve yesterday, and I'm not going to tell you exactly where, but I found a whole group of this white mushroom growing in the nature reserve, and it belongs to a species called Amanita bisporigera. Now, the common name of Amanita bisporigera is the destroying angel mushroom, because the toxins in A. bisporigera, which, by the way, you can't break down the toxins by cooking this mushroom. It's just as deadly cooked as raw. The toxins block RNA polymerase. They take this entire process of making messenger RNA and jab a monkey wrench right in the gears. They shut it down. This has the nasty side effect of making your liver die. Blood from your digestive system goes directly to your liver, and that's where any poisons in the food that you eat get broken down, but the liver is not able to handle the toxins in Amanita bisporigera. About 12 hours after you eat it, you start getting really ill, and then you feel better for a little bit, but then you just kind of collapse because your liver is going necrotic, and you die in an exceptionally unpleasant way over the course of several days. So I just thought I'd throw that in, because that does in fact grow on the UCA campus, and no, I'm not telling you where. And it actually blocks this process of transcription, making an RNA copy of DNA information with exceptionally unpleasant end results. Right. Okay. What happens next is a process called translation. Messenger RNA, if we're in a nucleus, passes out of the nucleus through a little pore called the nuclear pores. There's a bunch of them, nuclear pores, and goes out to a tiny structure called a ribosome. And you have tens of thousands of ribosomes floating around in every cell, probably more than that. Every ribosome is made of about 50 proteins stuck together. There's also yet another type of RNA that makes up part of the ribosome. It's actually called ribosomal RNA. Duh. We don't really need to get into that. We don't need to worry too much about what ribosomes are actually made of. Again, these are way too small to ever see with a light microscope. They're about 20 nanometers long. That's 20 billionths of a meter. The very tiniest things you could see with the best light microscopes possible would be about 500 billionths of a meter long. So this thing is less than a tenth the size of the smallest things you could ever see with light. We have to use electron microscopy to see these. And the messenger RNA makes like Madonna in 1983 and gets into the groove. It slips into a groove on the ribosome. Mute somebody, please mute. I'm still coming off a cold. Has whoever got your mic on, please mute. That's a computer model of what a ribosome looks like. And we worked it out through, well, among other things, quite a lot of X-ray crystallography. Remember that technique that lets you work out molecular shapes by zapping crystals of a molecule with X-rays. And we don't really need to worry about the details here, except it's made of two subunits. Let me circle them. One is this blob of proteins and structural RNAs that's over here. This. And the other subunit is this blob of proteins and structural RNAs that's over here. This. And you will notice that in between the two, there is a handy groove. Messenger RNA is able to recognize this groove. It's got a lead-in sequence that can do this. And the messenger RNA will slide itself into that groove in between the two subunits, almost like a piece of magnetic recording tape slid between recording heads. All right, checking the chat there to see what's what. Okay. So we've talked about messenger RNA that gets into the groove. There is a second type of RNA called ribosomal RNA that is just part of the ribosome, and we don't really need to worry about how that works. But the other type of RNA that you need to know about is called transfer RNA, or tRNA. And a tRNA molecule is a single strand, but it sticks to itself at several points, kind of like a piece of tape that you're trying to wrap a present with. You ever have that happen to you? You pull out a piece of tape and all of a sudden you find you can't use it because it has stuck to itself repeatedly. Transfer RNA molecules kind of look like that. It's a single strand, but, excuse me. But parts of the strand form hydrogen bonds with other parts of the strand. Sorry, guys, that was gross. And you end up with a molecule twisted on itself to form a set of loops. One of the loops is called the anticodon loop, and it just happens to have three bases that are sticking out in a prominent position, almost like buck teeth. Three bases that stick out, and one end of the transfer RNA is bound to an amino acid, which I hope you remember are the monomers that make up proteins. And which amino acid is present depends on the bases in the anticodon loop. So there's a simplified molecule of a transfer RNA molecule, simplified model, sorry, of a transfer RNA molecule. It's a single strand. It starts here and it ends here. And you can see there are 4 places where that molecule binds to itself to make a shape that's a little bit like a twisted clover leaf. And right here, this loop called the anticodon loop just happens to have 3 bases that stick out. And at one end of the transfer RNA molecule, there is an amino acid. And thanks to some magic worked by enzymes called aminoacyltransferases, which amino acid is up here depends on which 3 bases are down here. You know, a transfer RNA molecule doesn't carry just amino acid, just any amino acid. Which amino acid it carries depends on what bases it's got on the anticodon loop. And it's called the anticodon loop because when we read a piece of messenger RNA, we read the nucleotides 3 at a time. And every group of 3 nucleotides is called a codon. And every codon is associated with one and only one amino acid, except for 3 codons that are stop codons. Those 3 codons mean, okay, stop assembling the amino acids into a protein. Kind of like the period at the end of a sentence, they essentially mean stop here. So how does this work? Here I've diagrammed a ribosome. You can tell, I drew this, you can tell I am not really a master with Photoshop, but that's a ribosome made of 2 subunits and a groove in between. And on the right, we have a piece of messenger RNA that, like old Flat Top, comes grooving up slowly. That's a Beatles reference. And the messenger RNA slides into the groove on the ribosome, and I haven't shown it, but there's a leader sequence at the beginning of the messenger RNA that basically says, okay, start here. And so there would be various nucleotides, you know, up in this area that I'm not showing for clarity. But where translation starts, the first 3 nucleotides in this position are AUG. There's lots of transfer RNA molecules that are floating around, and various ones will fit into a slot on the ribosome to see if they can bond with the messenger RNA. But only one transfer RNA can do it, and that's the one that has UAC in that position. That's the only one that has nucleotides that are complementary to the AUG on the messenger RNA. And it just so happens that transfer RNAs that have UAC on the anticodon loop all happen to carry the amino acid methionine, which I've represented here as this green circle and just abbreviated as MET. That is the amino acid methionine. So all of the transfer RNAs might come in and see if they can fit. The only one that will fit is the one that's carrying UAC, which matches with AUG. And the transfer RNAs that carry UAC are the ones that carry the amino acid methionine. Now, the ribosome slides over 3 spaces, click, click, click. And the next 3 nucleotides happen to be UUU, right there. That's the next codon, the next grouping of 3. All those little transfer RNAs in the background are going to try to fit in. It's kind of like all of the princes bringing the shoes to see if, no, sorry, I've got it backwards. It's like all of the girls in the kingdom coming to see if their foot will fit the glass slipper. in Cinderella. But of all the ladies in the kingdom, the only one whose shoe fits the glass slipper is the one that's got AAA on the anticodon loop. Okay, I kind of jumbled my metaphors there. The only transfer RNA that can bind with this codon right here, these 3 nucleotides, the only one that'll fit is the one that's got AAA on the anticodon loop to bind with UUU. And the only transfer RNA molecule that has AAA on the anticodon loop is carrying the amino acid phenylalanine, which I've abbreviated PHE. Okay, now what? So the next 3 nucleotides are UUU. The tRNA that matches is the one that has AAA. And yes, the 2 stepsisters cut parts of their foot off to fit the shoe, and the prince was about to marry one of them until the birds came and said, "Look at the blood," and he noticed there was blood pouring out of the glass slipper, which is very unhygienic. But let's not go there. That's the fairy tale. That doesn't really apply to translation. Okay, the next 3 nucleotides are UUU. The only tRNA that matches is the one with AAA, which is a Canadian transfer RNA molecule, because it's all it says is A. Anyway, and that carries phenylalanine in. A peptide bond forms between these 2 amino acids, and the first transfer RNA molecule falls off. And eventually it will get another methionine stuck on it and get recycled, but we don't need to worry about that. What now happens is the ribosome moves down another 3 spaces, ka-chunk, 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 and you'll notice that now these 3 nucleotides happen to be UCA. And yes, our fair university is one of the few universities in America that is also a RNA codon. So, UCA is, stands, well, what does UCA stand for? Well, it stands for an amino acid. We ought to put that in our mission statement. There's not too many that also do, although, what was it? We used to play Abilene Christian University, and that's also a codon, ACU. And University of Georgia is UGA, and Catholic University of America, CUA, that also counts. So we do have some other universities that are also codons. But here we are, there's UCA right there. And all of the transfer RNAs will try to fit and see if their foot fits into the glass slipper. But the only one that will fit is the one that's got AGU on the anticodon loop right there. So the only institution that fits UCA is the American Geophysical Union, AGU. Okay, now my metaphors are getting really mixed. But yeah, the only transfer RNA that fits has AGU, and transfer RNAs that have AGU on this anticodon loop are also carrying the amino acid serine. So there's the amino acid serine. Serine forms a peptide bond with phenylalanine. So now we have 3 amino acids, one after the other. And the process continues on and on and on. Every time that happens, a new transfer RNA will come in. It will bring the right amino acid. The old transfer RNA molecule will fall off, and this will keep going until we have a whole chain of amino acids. And the order in which they go in has been read off of this messenger RNA, and it'll keep going until we finally hit a stop codon, which brings the process of translation to an end. The chain of amino acids that the ribosome has made floats away. The messenger RNA floats off and it might get reused or broken down. It depends. And we've assembled that chain of amino acids in the order specified by the messenger RNA. And the complete code book looks like this. So for example, first position's on the left, second at the top, third at the right. So this right here stands for UUU, which is the amino acid phenylalanine. UUA right here, UUA would be the amino acid leucine. There is UCA, UCA, that's the amino acid serine. Catholic University of America, CUA is also the amino acid leucine. Abilene Christian University, ACU is the amino acid threonine, and so on. You don't really need to know this code book, but that's how you read it. And there are 64 possible 3-letter combinations of bases. If you have 4 bases, there's 64 possible groups of 3 bases. 61 of them stand for one of the 20 amino acids that form proteins. 3 of them, UAA, UAG, and UGA, stand for stop codons. And those, again, basically say they don't stand for an amino acid. They say, okay, stop putting amino acids together, end translation. And that's what we call the genetic code. There you've got that code book. So once the ribosome hits that stop codon, the process is over. The completed protein falls off. The ribosome now separates into subunits, and the subunits will come back together and repeat the process. And this process is happening something like thousands of times a second in every one of your healthy cells. It's constantly going on. You're always making new proteins. And it's done by this process of copying information onto RNA, which is transcription, and reading it on the ribosomes, which is translation. You may have to modify the protein in various ways before you can use it. We call that post-translational modification. Even after you've put the amino acids together, you might have to stick other molecules to it, or maybe cut the chain in some way. We don't need to worry about that very much. And here we are at the top. This has been a lot. It may be a lot to absorb. But we started way back in the foothills with Gregor Mendel growing his pea plants, working out that there are these factors or elements, or what we now call genes, that are inherited from parents that produce the traits that he could see in his pea plants. Okay, so we worked out that genes are some kind of particle of inheritance. From then on, we worked out that genes are physically located on chromosomes. And then we worked out that chromosomes are long strips of DNA. And what a gene really is a length of DNA that's part of one of those long strips. That genes are physically located on chromosomes. We went from there and worked out that chromosomes are made of DNA. And then we worked out what shape DNA has. And we worked out that what's important is the order of nucleotide bases in a piece of DNA. And what I've covered today is the way that cells read the order of bases in your DNA and use that to assemble proteins. If you inherit a chromosome that's got a piece of DNA that codes for proteins with the right amino acid order, you'll make the right protein. If, on the other hand, you're like one of those guys that Archibald Garrod studied, whose urine turns black, sorry, I'm still stopped up. If you inherit this disease that makes your urine turn black, it's because you inherited DNA instructions for making a protein that are faulty in some way. The same is true if you inherited Duchenne's muscular dystrophy or Charcot-Marie-Tooth syndrome or cystic fibrosis or Marfan syndrome or sickle cell anemia or glucose 6-phosphate dehydrogenase disorder, or any one of a whole number of diseases that humans can have. If you have those, you have them because you inherited instructions for making a protein that, when they are read by transcription and translation, make a protein that's got the wrong order of amino acids and so doesn't have the right shape. That's the terrain that we've covered. Now, in the time that we've got, what can we see from the top of the mountain that we have just climbed? What's the view look like? Well, I'll start with a disease of tobacco plants. That's a tobacco plant, and it is diseased because the leaves have these light yellow patches on them. And this is a disease that tobacco farmers hate because it ultimately kills the plants. Tobacco plants that have this develop these irregular patches, and so the disease is called tobacco mosaic disease, right? A mosaic is a picture that's made out of tiles of different colors. So I guess someone thought that the leaves of a sick tobacco plant look like a mosaic because they're made of patches of different colors. Okay. Back in the late 1800s, people tried to figure out what was causing this, but nobody could find bacteria that were causing tobacco mosaic disease. And it's also not a genetic disease. And two folks, independently of each other, a Russian named Ivanovsky and a Dutch guy named Beijerinck, independently found that if you ground up the juice of an infected leaf and spread it on the leaf of a healthy plant, the healthy plant would get the disease. So there's something in the leaf juice that can make healthy plants sick. That's Beijerinck, by the way, in that portrait. The problem was that you could pass that juice through a filter that was so fine that it would strain out any bacteria that were present. And yet even the filtered juice would make the tobacco plant sick. So whatever is in that juice is smaller than any bacterium, and it's smaller than anything you could see with a light microscope. Beijerinck said, okay, we have to give a name to this mystery thing that is too small for us to see and too small to be a bacterium. And he called it a filterable virus, virus being one of the Latin words for poison. And we usually strip out the word filterable now and just call this unknown thing that causes tobacco mosaic disease a virus. By the 1930s, we developed microscopes that were powerful enough to get pictures of the tobacco mosaic virus for the first time. It turns out that it's, remember, a micron is 1/millionth of a meter. The tiniest things you can see with a light microscope are 0.5 microns. A TMV particle is 0.3 microns long. It actually looks like a tiny cigarette, which is funny to think that tobacco gets sick from cigarettes. I don't know, kind of cute. It's not made of cells, and it does contain proteins and nucleic acids, but it's got no metabolism at all. It's completely inert. You could take a jar of tobacco mosaic virus and hook it up to machines, and it would do absolutely nothing. It doesn't breed, it doesn't feed. In fact, you can even make crystals out of it. So what the heck is this thing? Well, it's a virus. Okay, what are viruses? Now we can finally reveal the truth. You know, one particular virus has been gripping the attention of the world ever since spring break 2020. Another much milder virus is still inhabiting my nasal passages. It's just a cold, it's not COVID. But you've heard of viruses a lot, but now we can finally get a look as to what they really are. A virus is a piece of DNA or RNA. It can be either, but it's never both, surrounded by a coating of proteins that fit together called a capsid. Now, this is not cell structure because viruses are not cells, because viruses don't carry on metabolism. Viruses cannot live on their own. In fact, viruses are kind of somewhere in the gray area between alive and not alive. They act like living things in some ways, but not in all. So if you remember that list I gave you way back at the start of this semester of the features of living things as we know them, viruses don't check all the boxes, but they do check some. So, you know, it's a, nice long philosophical question as to whether you should call these living or not. So viruses are neither considered prokaryotes nor eukaryotes. They're not considered cells. So that prokaryote eukaryote distinction doesn't even apply. And in isolation, viruses are completely inert. They do nothing. But if a virus comes into contact with a host cell, the virus will inject its DNA or its RNA into the host cell, and a cell that's infected will start copying the virus's DNA or RNA, and it will start transcribing and translating the virus genes to make more virus proteins. And the proteins and the virus DNA or RNA assemble inside an infected cell. So a virus turns a cell from a factory for making proteins for its own use into a factory for making more viruses. You might remember computer viruses. They actually work in an analogous way, in that if your computer gets a virus, what the virus does is start sending out copies of itself to other computers, which it might do over the email network or over the internet, or I guess nobody shares infected floppy disks these days, but they used to a long time ago. What a computer virus does is turn your computer from a machine for writing your term papers or playing, I don't know, God of War or whatever it is that you do. It turns that computer into a machine for making more copies of the virus and spreading them to other computers. Biological viruses work in an analogous way. So that's what one particular virus looks like. The virus's capsid is made of proteins that pack together, often in a very regular and symmetrical way. Sometimes they look like soccer balls. This particular one happens to be the Zika virus, This is one that we were freaking out a few years ago, back before COVID, back in the before time, before we were worried about coronavirus, we worried about Zika, because Zika is transmitted by mosquitoes and it has the potential to cause birth defects in the fetuses of infected women. Every type of protein here is shown in a different color. So you have small blue proteins sticking out of a sphere that's made by the packing together of these orange, red, and yellow proteins. And if you look carefully, you can see that the result is actually quite geometrical. Many viruses are quite beautiful. So yeah, that's a virus capsid. And in life, it's a sphere. And inside there is the Zika virus's, I can't remember if it's DNA or RNA, but that's where the Zika virus's nucleic acid would be. Oh, Roz Franklin's other major accomplishment, aside from working out the double helical structure of DNA, was that she used X-ray crystallography to work out the basics of tobacco mosaic virus structure. That in itself would have been worthy of a Nobel Prize. And it's really a shame that she died young, because I think that was the first, that may have been the first virus whose structure got worked out in that kind of fine detail. What you're looking at here is in yellow, those are capsid proteins of tobacco mosaic virus. And when they fit together, they make a cylinder. Inside the cylinder, you have nucleic acid. In this case, it happens to be RNA. It's shown in red, and it forms a coil that fits inside the capsid. So the capsid here has been partly, cut away to show you how the RNA fits inside. This, by the way, happens to be a virus that infects bacteria. It's called a bacteriophage. And on the right, you can see there's a computer graphics reconstruction. The nucleic acid is kind of rolled up in here. That's it. And then the main part of the capsid is, has this really cool geometric shape. And then it's got a shaft and a little base plate and legs on it. So it almost looks like a lunar lander. And so there's a diagram of it on the right, and on the left, those are some actual photos taken with an electron microscope. So we can see these viruses now. That bacteriophage has attached itself to a host bacterium, and we've caught it in the act of injecting its DNA directly into the host. This is the only thing that viruses can do. They'll just drift until they happen to encounter a host cell. And if they encounter a host cell, they can latch onto the host cell and inject their DNA right in. That's what the bacterium looks like a few days later. This bacterium is now packed full of virus capsids. It's full of bacteriophage. It's full of bacteriophage particles. These are assembling inside the bacterial cell, and they eventually burst out and kill the cell and go on to infect other cells. By the way, and I mention this because I've got a friend of mine that is undergoing this. Viruses are usually very specific for only one type of host cell. Bacteriophage viruses cannot infect eukaryotic cells. Bacteriophages cannot hurt you. You could drink a glass of them and they would not affect your own personal cells. That means that as far back as the 1930s, there have been experiments made to grow bacteriophage viruses and use them to treat bacterial infections in humans. Instead of trying to poison the bacteria with antibiotics, you could stop bacteria growing in you by taking bacteriophage that would invade the bacteria that are making you sick and make more viruses and kill the bacteria in the process. It does sometimes work, and a friend of mine is actually undergoing phage therapy for pneumonia that she's got that is resistant to all antibiotics that we have. It's still, I don't want to say experimental, it's still kind of unusual, and I'm not sure that therapy is available in the United States, but it may end up coming into wider use as more and more bacteria develop antibiotic resistance. So people have used this as a way to try to kill disease-causing bacteria, which I think is kind of cool. So yeah, that picture shows a bacterium going explodey, as my friend says, releasing more phages out into the open. Okay. And viruses work the same way in eukaryotic cells. They replicate inside and then come bursting out. Some viruses will take little pieces of the host cell membrane with them. You're looking at pictures of a human herpes virus. One type of herpes virus causes genital herpes. There's another type that causes cold sores. And what you're looking at is the capsid is right there. And each one of these little dots that you see is a single protein. We can actually see single protein molecules if we use the right type of microscope. But wrapping around the whole thing is a piece of the host cell membrane that the herpes virus is torn off and is taking with it. Kind of like Buffalo Bill in Silence of the Lambs. It's torn off a piece of its victim and wrapped itself in it. Ew, yuck, sorry. You see the same thing on the right. That's a capsid right there. But then surrounding the capsid is a piece of the host cell. This is one of the things that makes herpes kind of tough to treat, is it's hard for your body to recognize herpes virus because herpes virus uses pieces of your own cells as camouflage. So that's one of the reasons why herpes is chronic. And there, by the way, is coronavirus. And coronavirus is wrapped in cell membrane, but on the outside of the membrane, you have spike proteins that are sticking out. In that computer graphics reconstruction, they're shown in red. They're also called fusion proteins, and they make the coronavirus kind of look like a crown if you're on drugs. Well, kind of. I mean, right here in this picture, that's an electron microscope picture of a coronavirus, and you can see the little spikies there, there, and there, and there, and there. So yeah, kind of looks like a, kind of looks like a crown. And since a crown is called corona in Latin, that's why they're called a coronavirus. So that's why they have that name. There you've got a bunch of coronavirus particles in an infected cell. This is a very thin slice of a cell magnified hundreds of thousands of times through an electron microscope. And every one of these little circles that you're seeing right there is a new coronavirus. And like all viruses, coronaviruses take over a cell's ability to transcribe and translate to make proteins and use that to make copies of themselves. So an infected cell can't make the proteins that it needs because it's using all of its energy to make coronavirus proteins. And coronavirus particles assemble inside a cell and eventually burst out, killing the cell, and go on to infect other cells. And this is what happens when you get infected with coronavirus. It's the same basic thing if you get a cold or the flu or smallpox, except theoretically nobody should get that anymore, or yellow fever, or Ebola, or chickenpox, or any one of a number of other virus diseases that people and animals can get. What is happening is the virus is taking over cells and using those cells to make more copies of themselves rather than the proteins that they need. Okay, how does the virus take over the cell? Well, it's just that the viral nucleic acid gets transcribed and translated at the expense of the cell's own DNA. More details than that, I'm not going to get into, and this is not what I am professionally specialized in. I will see if I can come up with a better answer for you. Hope that'll do for now, but I'll see if I can give you a better answer than that on Thursday. Little bit on COVID. I haven't talked about this much, but cells can sense the environments that they're in, and they can respond to those environments. And they do that because they've got proteins on their surfaces. You can kind of pretend that these proteins are little antennas that can pick up other molecules in the cell's surroundings. Cells can, some of them can move in particular directions because they can pick up chemicals around them and move towards them or away from them. Cells have all kinds of receptors on the outside. And in humans, one of the receptors that your cells have just happens to be called ACE2, which is what enables your cells to respond to a hormone called angiotensin that regulates blood pressure. The way this has been diagrammed, every one of these things that looks like a little stick is actually an alpha helix. So these gray, purple, blue, and pink areas right there, each one of those is a separate polypeptide. And then all four of them fit together to make a shape that looks a little like a kind of blobby X. So that protein right there is the way that your cells can respond to a hormone that your body makes. And part of the protein sticks out of a cell, I think like the cell membrane goes here, and part of the protein sticks out and part of it is inside. There's a lot of these receptor proteins that actually cross the cell membrane, like a TV antenna. You don't even remember those. Okay, like a satellite dish is partly on the roof, but it's attached to wires that go inside your house. There we go. And what you're looking at here is a computer graphic picture. You've got a host cell is down here, and each of these blue things is one of these ACE2 receptors. So in the lower part of this picture, this is a diagram of a host cell. Up here at the top, this is a coronavirus particle, and in pink, these things are the spike proteins, and coronavirus is able to recognize your cells because its spike proteins have the right shape. There's protein shape again, that they bind to ACE2. They've got just the right shape to bind to ACE2. Once that's happened, the virus is stuck to the outside of the cell, and the virus can penetrate into the cell. And then, by the way, in the distance right here, this thing right here, this big blob, that's a COVID virus particle that is in the process of bursting out. Kind of like all the cold viruses that are bursting out of the cell of my nose. Fortunately, since we're remote.
Gesundheit.
Thank you. Fortunately, since we're remote, you can't, you're not going to get these. Sorry, I'm kind of a snot rocket this morning. I thought I was doing better. By the way, I'll talk more about this on Thursday. Your body defends against viruses with a population of cells called cytotoxic T cells. In this picture, a T cell has been colorized. It's not, these are false colors, but the orange blob is a cytotoxic T cell that is destroying a cell that's gone cancerous. And cytotoxic T cells can also destroy virus-infected cells. So this is part of your body's defense against viruses. Excuse me. Yuck, sorry. It's also part of your body's attempts to defend against cancer. What this picture is showing you is HIV. Human immunodeficiency virus is another cell that wraps itself in cell membrane. On the left right here, this is a host cell. And all of these little blobs right here are HIV virus particles that are budding off of an infected cell. And what makes HIV dangerous is it attacks immune system cells called CD4 cells, sometimes called helper Ts, that help those CD8 cells that you saw earlier. So what makes HIV devastating is that it's a virus that goes after the cells that are involved in defending against viruses. It would be the equivalent of the mafia taking over the police, right? The police are supposed to keep you safe from criminals, but if the criminals go after the police, who's going to keep you safe? That's essentially what HIV does. It directly attacks your ability to defend against virus infections. And antibiotics do nothing against viruses because they attack bacteria. Penicillin blocks some bacteria's ability to make cell walls. That's a function of metabolism. Viruses don't have metabolism. So taking an antibiotic when you've got a cold does absolutely nothing, except maybe the placebo effect, if you remember what that is. There are some drugs that we can use. This happens to be a drug called interferon. And you can see it's made of a lot of alpha helices. It's a protein. Excuse me. And it stimulates the immune system to step up its game against a virus infection. Excuse me, I'm going to blow my nose. All right, sorry about that. It's kind of gross. So yeah, I'm shedding viruses right now, although fortunately not to you. And the last thing I'll mention, because it ties in with all this stuff about DNA and RNA that we've talked about, is that there are cutting-edge antiviral drugs that look like nucleotides, but are just different enough that they can't be used to make nucleic acids. So if a cell tries to use these, if you fill a cell full of drugs like this, the cell will try to use them in either replicating DNA or transcribing, transcription to make RNA, and the process will get blocked. Kind of like if you have a stapler and you feed it staples that are misshapen, the entire stapler gets jammed. That ever happened to anybody? You ever get a jammed stapler? It's the same thing here. You can jam the enzymes that carry out transcription or carry out DNA replication if you feed them drugs that look like nucleotides but aren't. So up at the top, that's a nucleotide called guanidine phosphate. That base right there happens to be guanine, that double ring of carbons and nitrogens. The sugar is deoxyribose, and there's the phosphate right here. If you feed a cell a drug called acyclovir, it's got the base right here, but instead of having the sugar, it's got nothing right there. And so if you pump a cell full of acyclovir, it will try to use this in DNA replication and fail. So acyclovir is the drug of choice for treating the herpes virus, by the way. It never completely gets rid of it, but it can knock it down to low enough levels that you can live with it. This is one that is in clinical trials for effectiveness against coronavirus. It hasn't been approved yet, but they're looking at it. This is a weird nucleotide, a real nucleotide here. The base is adenine, and there's deoxyribose, and there's phosphate. Galidesivir has adenine, and instead of ribose, it's got this weird sugar that's got an extra nitrogen in it right here that's not supposed to be there. Galidesivir was developed to treat hepatitis C. It seems to be effective against Ebola, and it's in clinical trials for effectiveness against coronavirus. There is another drug that's been in the news called remdesivir that was tried in the early days of the epidemic. It doesn't seem to be effective against everybody, but I think it still can help in severe cases. So it's under, I think it's authorized for emergency use right now. Remdesivir actually works the same way. It's also a fake nucleotide that a virus-infected cell will try to use to replicate virus DNA or RNA. And what happens is that the works get jammed and the viruses don't replicate. Here's another one. This one's sold under the trade name Videx. This right here is adenine up here at the top, which connects to a sugar, deoxyribose, and a phosphate right there. Videx has this weird base called inositidine, which you haven't seen before. But more importantly, it's got a sugar with no oxygens on it. This sugar is called dideoxyribose. It's missing a phosphate, but that's not important. Your body can easily stick one on. That happens all the time. But with no oxygens on it, it can't link to any other nucleotides. So if you feed a cell that's making virus DNA or virus DNA full of Videx, the strands can't extend any further and the cell can't make any more virus particles. Videx, by the way, is one of the drug cocktails that's used to treat HIV. And there's other drugs like this that are effective against things like cancer, where cancerous cells are also transcribing and translating and replicating DNA and RNA at a very high rate. And yeah, they interfere with normal cells too, which is why these drugs have side effects. This is why you can't buy them over the counter. They will poison you if you take too much or in the wrong way. But the side effects are outweighed by the beneficial effects of stopping viruses. So the HIV cocktail, when I was your age in the 80s, good God, that wasn't that long ago, they were only beginning to come up with drugs like this, and people would tend to take just one at a time. What they do now is, if you have HIV, you have to take enough pills to make you rattle, but they hit you with several drugs at once. They'll give you some of these fake nucleotides like Videx that block transcription. There's other drugs that interfere with fusion proteins that help the virus bind to the cell. There's others that block the virus from assembling. There's others that block new viruses from breaking out of the host cell. So if you have HIV, they try to hit you with several weapons at once. And that's more effective than just one approach would be. And you can use these to treat other viruses. So Tamiflu, for example, which I am told sucks, and you should get something called Exafluza. Thank you for the tip. Those block flu viruses from breaking out of the cells. So if you know your transcription and translation, you can develop different strategies for combating virus infection. And that would seem to be it, and we're just about out of time. So by the power vested in me by the state of Arkansas, I will stop recording